Hello guys, welcome back to my channel. This is the Cortex and today we will be continuing with IGCSE CAIE biology, and the chapter is diffusion and osmosis and surface area. In this topic we will be covering active transport as well. Now this is our learning objective. This is part 1. So in this we will learn diffusion, factors affecting the rate of diffusion, osmosis, hypertonic, hypotonic and isotonic solutions, as well as plant cells in high and low water potentials, plasmolysis and turgor pressure, and importance of turgor pressure in plants. If you want to see these topics, we will cover those in part 2. So let's get on with it. Diffusion. Now what is diffusion, one might ask. Diffusion is the net movement of ions or molecules from a region of higher concentration to a region of lower concentration down a concentration gradient. First of all, you can see in this picture that here is the region of higher concentration of molecules, and here we see that it starts to diffuse across the lipid bilayer, and here both there is a same concentration on both sides of the cell membrane or the lipid bilayer in the extracellular and the intracellular spaces. In liquids and gases, molecules possess kinetic energy and constantly move about, which we also learned in our chemistry class. As this movement is random, an equilibrium is reached when the molecules are evenly spread out, where there is no more net change. Dynamic equilibrium is reached. Now equilibrium we will study it about deeply in physics, but just remember that it is most like equality. The steeper the gradient for a substance, the faster the rate of diffusion. Now this you see over here is the concentration gradient. It basically is this slope or graph. We see over here that this the y-axis shows the concentration of the molecules, and this shows basically the diffusivity. And see, you can see that there was first high concentration of water molecules and then it spread into low concentration of water molecules. No energy input is required for diffusion because the particles are already in motion. Hence it is known as passive transport. It is passive because it does not need any sort of energy. See, this is a great example. Over here in the first picture we have 20 potassium iodide ions, and in the second we have 20 copper sulfate ions. So they will obviously diffuse because in over the second part there is not any potassium iodide ions, which means lower concentration over here and a higher concentration over here, and vice versa with the copper sulfate ions. So they all start to diffuse until equal amounts of both are left on both sides. See, 10 potassium iodide ions and 10 copper sulfate ions on both sides. This is obviously a permeable membrane, which permeable meaning it can let anything go through it and come from it. Factors affecting the rate of diffusion. Obviously first thing we will remember is the concentration gradient, that how quickly these diffuse. The temperature, like you remember we learned in physics that diffusion is affected by temperature. Pressure, again we learned in chemistry, temperature as chemistry also. The size of the particles, obviously some particles can go through the membrane, that's why the cell membrane is known as a partially permeable membrane. And the surface area to volume ratio, which we will be discussing in a minute. Osmosis. Now this is the second process and this is very important. Osmosis is the movement of water molecules from a solution of higher water potential. See, here we use water potential, not concentration. Please do not write concentration because it will make no sense. To a solution of lower water potential through a partially permeable membrane down the water potential gradient. Again, not a concentration gradient but a water potential gradient. Water potential is a measure of the tendency of water to move from one place to another. Basically the concentration of water molecules is known as water potential. A water potential gradient is established when a partially permeable membrane separates two solutions of different water potentials, because a water potential gradient and concentration gradients are formed when there is a partially permeable membrane so that both can diffuse. A dilute solution, which means higher amount of water potential, and a concentrated solution means lower water potential, contains more water molecules per unit volume than a concentrated solution. See, it says here, so it has a higher water potential than a concentrated solution. Again, osmosis. Osmotic pressure is the pressure that must be applied to a solution to prevent the inward flow of water across the partially permeable membrane. So this is osmotic pressure. Osmotic pressure is the opposite of water potential. A partially permeable membrane is a membrane that will allow certain molecules or ions to pass through it by diffusion, which we learned in our first lesson of biology. The cell membrane is partially permeable. We see it's partially permeable. I told the hydrophobic and hydrophilic tails and heads. So water does not pass through here, but it has a special protein known as aquaporin, which facilitates in the diffusion of water molecules, or osmosis. This is a great example. Also, in one side we have 8 sucrose molecules and 9 water molecules, and the other side we have 4 sucrose molecules and 18 water molecules. You can see that the ratios and the amount is not same. So what will happen? These will diffuse, and here is the semi-permeable membrane or partially permeable membrane. And now 8 to 18 and 4 to 9, you can see that the ratio is same, because if 4 to 9, if we double that ratio, it will be 8 to 18. That is osmosis. Ratio is considered very important. Hypotonic, isotonic and hypertonic cells or solutions, there can be both. Now we see a hypertonic solution. It means, hyper means a high concentration, which means lower water potential. We see it says plasmolysis, which we will learn in this video. But basically what happens, because there is a less water potential, the osmosis will carry on to make it the same water potential, which we learned in diffusion and in osmosis. So the cell will release its water molecules. But whereas in hypotonic, to make there is a higher water potential outside the cell, and inside is a lower water potential, so the water molecules from outside will enter the cell so that it can be an isotonic solution. Now what is an isotonic solution? Where the water potentials in the extracellular space and in the intracellular space of the cell are equal. Plant cell in high water potential. Water enters the cell as the cell is hypertonic, which means a lower water potential. Hypertonic. It will be hypotonic over here. I'm very sorry about that. The vacuole increases in size and pushes the cell contents against the cellular cell wall. The cell wall prevents over expansion of the cell by exerting an opposing pressure, preventing the entry of more water. I'm very sorry, it will be hypotonic over here, which means that there is a higher water potential outside the cell. So to equalize or to reach equilibrium, the water will enter inside the vacuole of the plant. And then the vacuole will increase size, which will put pressure onto the cell wall, which will also again put pressure onto the cell, and this is turgor pressure. A plant cell in this stage becomes turgid. The turgidity of the cell with water is called turgor pressure. The plant cell does not burst because the cell wall is strong and relatively inelastic. Plant cell in low water potential. When a plant cell is placed in a solution with low water potential, it means it is placed in a hypertonic solution. Water leaves by osmosis and the cytoplasm shrinks away from the cell wall. This is known as plasmolysis, and now the cell is plasmolysed. This state of the cell tells us that this is flaccid. See, when it is put in, it will start to equalize the water potential, so it will release all its water molecules. That's why it will shrink in size, and this state is known as the cell has become flaccid due to plasmolysis. This process is known as plasmolysis. Now, the importance of turgor in plants. Turgor, you know, the push. Turgor is very important in maintaining the shape of soft tissues in plants. Young stems and most leaves, especially those of herbaceous or non-woody plants, can remain firm and erect because of the turgor pressure within their cells. Because of the turgor pressure, they put the pressure on the cell walls. That's why they can stay firm and erect. Unlike a plant which is flaccid, it will start to wilt, what people will say, and because of the enlarged vacuole, it will stay firm. When there is a high rate of evaporation of water from the cells, they lose their turgidity and the plant wilts when it is flaccid. The movement of certain plants are due to changes in turgor pressure. For example, opening and closing of the stomata, which we will also learn in the chapter nutrition in plants, and transport folding of leaflets of the mimosa plant, when that is also due to turgor pressure. Thank you. It has been a pleasure. Please don't forget to subscribe and like this video. Thank you.